Ribosomes are tiny molecular machines found in all living cells. They are responsible for one of the most important processes of life, protein synthesis. Ribosomes read the genetic information carried by messenger RNA and proteins. Therefore, the ribosome is often called the protein factory of the cell. In prokaryotes, ribosomes occur throughout the cytoplasm. In eukaryotic cells, ribosomes may be free in the cytosol or attached to the membrane of the rough endoplasmic reticulum. The ribosome was discovered in chemical and microscopic studies of cells during the 20th century. Researchers found small particles associated with the cellular.
George Emile Palade played a major role in identifying these small cytoplasmic particles and demonstrating their association with the endoplasmic reticulum. Later research established that these particles are responsible for protein synthesis and revealed their complex RNA-protein architecture.
Structurally, a ribosome is a ribonucleoprotein complex composed of ribosomal RNA and numerous ribosomal proteins.
The small subunit primarily helps decode the messenger RNA, while the large subunit contains the catalytic center responsible for peptide-bond formation.
Together, the two subunits create a highly organized molecular environment where mRNA and transfer RNAs interact during translation.
The ribosome contains three important functional regions, the A site, P site, and E site, which coordinate tRNA entry, peptide chain holding, and tRNA exit. Prokaryotic cells, such as bacteria, contain 70S ribosomes in their cytoplasm. The bacterial 70S ribosome consists of a 50S large subunit and a 30S small subunit. The 30S subunit contains 16S ribosomal RNA and approximately 21 different ribosomal proteins. The 50S subunit contains 23S and 5S ribosomal RNAs, together with numerous ribosomal proteins. In standard bacterial descriptions, the 30S subunit contains 21 proteins and 16S rRNA and 5S rRNAs. Eukaryotic cytoplasm contains larger 80S ribosomes, either free in the cytosol or associated with the eukaryotic 80S ribosome consists of a 60S large subunit and a 40S small subunit. The 40S subunit contains 18S ribosomal RNA and approximately 33 ribosomal proteins. The 60S subunit contains three major rRNAs, 28S and 5S, together with many ribosomal proteins. The 40S subunit contains about 33 proteins, while the 60S subunit contains about 47 protein types in the simplified composition shown here. Ribosome assembly begins with the synthesis and processing of ribosomal RNA, together with the production of ribosomal proteins. In the small subunit pathway, 16S rRNA folds and interacts with specific ribosomal proteins. This process for the large subunit, 23S and 5S rRNAs associate with ribosomal proteins to form the developing 50S subunit. After maturation and quality control, the functional 30S and 50S subunits join during translation to form the 70S ribosome.
In eukaryotic cells, ribosome biogenesis is a highly coordinated process involving the nucleus.
Ribosomal RNA is transcribed and processed in the nucleolus, where it associates with ribosomal proteins imported from the cytoplasm. Immature ribosomal subunits undergo extensive processing and quality control before being exported through nuclear pores.
Once mature, the 40S and 60S subunits participate in translation, forming the functional 80S ribosome.
The central function of the ribosome is translation, the conversion of genetic information encoded in mRNA into a specific. Translation begins when the ribosome recognizes the mRNA and positions the start codon correctly for initiation. An initiator tRNA pairs with the start codon, and the large subunit joins to form the complete functional ribosome. During elongation, amino-acyl tRNAs enter the A site, while the growing peptide chain is held in the P site. The ribosome catalyzes peptide-bond formation. Remarkably, the catalytic center is formed primarily by ribosomal RNA. After peptide-bond formation, the ribosome moves along the mRNA. This process, called translocation, advances the message by one codon. These cycles repeat rapidly, adding amino acids one by one and producing a growing polypeptide chain according to the mRNA sequence. When a stop codon reaches the ribosome, a release factor recognizes the signal and promotes release of the completed polypeptide. Free ribosomes generally synthesize proteins functioning in the cytosol and certain organelles, whereas ribosomes bound to several ribosomes can translate one mRNA simultaneously, forming a structure called a polysome, greatly increasing protein production. Thus, the ribosome performs several coordinated tasks: mRNA decoding, tRNA positioning, peptide-bond formation, translocation, and accurate termination. Ribosomes are essential molecular machines that connect genetic information with cellular function by converting mRNA instructions into proteins. From their ribosomal RNA core and protein components to their A, P, and E sites, every structural feature contributes to accurate and efficient translation. Therefore, the ribosome is not simply a cellular particle. It is a sophisticated RNA protein machine that lies at the heart of protein synthesis and cellular life.